Session 12, first semester, BSc, University of Mysore. The topic of the session is classification of bacteria based on nutrition. Bacteria require about 10 elements in large quantity, that is macronutrient, because they are used to construct carbohydrates, lipids, proteins and nucleic acids. Several other elements are also needed in very small amount and are parts of enzymes and cofactors. Among these, the elements which are required in large quantities are called macronutrients. 95% or more of cell dry weight is made up of a few major elements, that is carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, potassium, calcium, magnesium and iron. These are macronutrients which are required in little large quantity for the nutrition of bacteria. The first six, CHONPS, carbon, hydrogen, oxygen, nitrogen, phosphorus and sulfur, are components of carbohydrates, lipids, proteins and nucleic acids. In addition to this, there are trace elements which are required for the microbes in very small amount of other mineral elements such as iron, copper, molybdenum and zinc. These are referred to as trace elements or micronutrients. Most are essential for activity of certain enzymes, usually as cofactors. In addition to this, there are some growth factors which are necessary for bacterial nutrition, that is amino acids, purines, pyrimidines and vitamins. These amino acids are needed for protein synthesis, whereas purine and pyrimidines, they are the components of nucleic acids and needed for nucleic acid synthesis. And vitamins are small organic molecules that usually make up all or part of enzyme or cofactors, and only very small amounts are required for their growth, bacterial growth. Therefore, bacteria are classified based on the nutrition into the following types, or this is also called as nutritional types of bacteria. The major nutritional types are, if you say, looking into this, that the photoautotrope, this is also called as photolithotropes, photo, light, self, auto, tropes, the tropic, that is synthesis. For this group of bacteria, photoautotrophs, the source of energy is light, and inorganic hydrogen has an electron donor, and also carbon dioxide is the energy source. And the example for this group is algae, purple and green bacteria, cyanobacteria. The second group is Photoheterotropes. They are also called Photoorganotropes. Normally, heterotrophs, they needs light. Such group of bacteria, their source of energy is light, and inorganic hydrogen is electron source, and carbon is from organic carbon source. Example of this group is purple non-sulfur bacteria and green sulfur bacteria. The third group of nutritional types of bacteria are chemoheteronotropes. They are also called as chemolithotropes. Chemo is chemical. The source of energy is chemical energy, that is from inorganic source. The source of electron is from inorganic hydrogen donor, and carbon dioxide is the source of carbon. Representative organisms are sulfur oxidizing bacteria, hydrogen bacteria, and nitrifying bacteria. And fourth group of nutritional types of bacteria are chemoheteronotropes. These are also called as chemoorganotropes. For this group of bacteria, the source of energy is chemical energy is the source, that is organic chemical source. And hydrogen is from organic electron donor. Carbon is from organic source, that is organic carbon. Example is the most of the bacteria, most of the fungi and protozoa coming under this group chemoheteronotropes. And these organisms like photoautotrope, like cyanobacteria and algae, they react with carbon dioxide plus hydrogen, that is the source is from the water. In presence of light and chlorophyll, they synthesize carbohydrate releasing oxygen. The purple and green bacteria, they are also carbon dioxide with hydrogen sulfide with the source of light and bacterial chlorophyll, synthesizes carbohydrate and water plus 2 molecules of sulfur is released. Whereas the photoheterotropes, example is purple sulfur bacteria rhodospirillum. In this case, the reaction with carbon dioxide and an organic compound reacts with the presence of light and bacterial chlorophyll, synthesizes carbohydrate. Bacterial photosynthetic system has certain properties which are studied in cyanobacteria, green sulfur bacteria and purple non-sulfur bacteria. Properties like photopigment. In case of cyanobacteria, this photopigment is chlorophyll. In green sulfur purple bacteria, it's a bacterial chlorophyll. In purple non-sulfur bacteria, that is also bacterial chlorophyll. The second property is oxygen production. Cyanobacteria will not produce oxygen. Green and purple bacteria produces. Cyanobacteria produces oxygen. Green and purple bacteria and purple non-sulfur bacteria is not producing oxygen. Third property is electron donors. Cyanobacteria, the source of electron donors is from water. In green and purple bacteria, hydrogen and hydrogen sulfide and sulfur. Even in purple and non-sulfur bacteria, hydrogen, hydrogen sulfide, and sulfur. The property like carbon source is carbon dioxide for cyanobacteria. Green and purple bacteria is also carbon source is carbon dioxide, whereas for purple and non-sulfur bacteria, carbon source is organic or carbon dioxide. The property like the primary product of energy conversion is, in case of cyanobacteria, ATP plus NADPH. In case of green purple bacteria, ATP is the primary product of energy conversion. That is also same in case of purple and non-sulfur bacteria, primary product of energy conversion is ATP. Some nitrifying bacteria, with the help of ammonia plus oxygen, they produce nitrate, releasing 132 kilocalories of energy. On the other hand, artificially, we can provide nutrition to the bacteria through cultural media, which are needed to grow microorganisms like bacteria in the laboratory and to carry out specialized procedures like microbial identification, isolation from water, food analysis, and isolation from particular microorganisms. A wide variety of media is available for these and also for other purpose. So, microorganisms can be isolated using cultural media. Coming to the importance, bacteria are economically important as they are used by human for many purposes. The beneficial use includes production of traditional food such as yogurt, cheese, vinegar. They are also important in agriculture for the compost and fertilizer production. They are used in medicine for the production of antibiotics, insulin, and many such. Helpful to degrade dead animals and plants and bring valuable nutrients back to the soil. And some species also clean harmful pollutants of environment by bioremediation. So therefore, for bacterial nutrition, amino acids are needed for protein synthesis. Purine and pyrimidines are needed for the synthesis of nucleic acids. Vitamins are small organic molecules that are usually make up all or part of enzymes as cofactors, and only very small amount are required in the nutrition for the metabolism of bacteria.